Gene knockout is a common technique. Therefore, these experiments are usually restricted to well-funded labs or core facilities. This has presented a large hurdle for researchers exploring gene function. The breakthrough of CRISPR technology has turned the tide and made gene knockout a much more accessible tool for many more research labs. A key revolution brought about by CRISPR is the creation of double-stranded breaks sequenced specifically. With CRISPR, scientists can create double-stranded breaks at virtually any site in the genome. To repair the double-strand breaks generated by CRISPR, there are two main mechanisms: non-homologous end joining and homology-directed repair, also known as homologous recombination. With non-homologous end joining, the ends of the double-strand breaks are simply reconnected together, and this process is highly error-prone, introducing mutations, insertions, and deletions, and in many cases resulting in gene knockout. Homology-directed repair, on the other hand, utilizes a repair template, where desired changes are flanked by left and right homologous sequences. Through homologous recombination, the desired changes are incorporated into the genome. The CRISPR knockout knock-in kits utilizes the homology-directed repair system. To plan an HDR-backed knockout experiment using plasmids, the researcher needs to create the following constructs. 1. A plasmid coating for the gRNA, targeting the intended site for double-stranded cleavage. 2. A plasmid coating for the Cas9 protein. And 3. A plasmid serving as the repair template, with a designed sequence flanked by the homologous arm region of the cleavage site or donor vector. Origin has created pre-designed kits to make knockout experiments even easier. For each gene locus, we have created a specific kit. The first two elements are provided in a single all-in-one vector. The gRNA vector, targeting the start of the coding sequence, was cloned into a vector containing the expression cassette for Cas9 protein. The donor vector was also created with a knock-in GFP puromycin cassette, flanked by the homologous arms of the chosen cleavage site. This permits the selection of knockout cells via puromycin marker. The promoterless GFP provides additional utility to monitor any upstream promoter activity that might be present. To start your knockout experiment, simply co-transfect a gRNA vector with a donor DNA plasmid into the cells. The Cas9 enzyme will cut a double-stranded break near the start codon, and through homologous recombination, the functional cassette will be integrated into the genome, replacing target gene. In the end, 3 events are achieved. 1. The target gene is knocked out through gene disruption. 2. GFP will be driven by the endogenous gene promoter, providing added information of promoter strength. And 3. The puromycin marker under the PGK promoter is integrated into the genome, enabling the selection of successfully edited cells. The protocol starts with co-transfection. There will be 3 separate experiments. One of the 2 gRNA vectors co-transfected with the donor vector, scramble control with donor vector. After transfection, the cells must be passaged for about 20 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin resistance. Culturing the transfected cells for 20 days ensures that the episomal form of donor vector is lost. This diagram shows the details of transfected cell passaging prior to puromycin application and selection. For HEK392 cells, 48 hours post-transfection is considered G1 cells, at which point the cells are split 1 to 10 and grown for 3 more days. At G2, CRISPR-targeted cleavage and gene editing should be finished, but the percentage of edited cells will be small. You can perform optional genomic DNA analysis using G2 cells. If the data are negative, you can redo the analysis after puromycin selection. After G7, apply puromycin selection using the doses appropriate for your cells. To summarize the CRISPR knockout knock-in protocol described so far, Step 1. Co-transfect one of the gRNA vectors with the donor vector. Along
1
with the
लाख
appropriate
गुरुद्वारे में हॉल
controls.
के लिए
Step 2.
घर में उसको
Passage
500 रुपए के लिए
transfected
कामारी
cells
बाई को
for
बदलते
around
देखा,
20 days. Step 3.
जिसने
Apply
दे दी माँ
puromycin
बाप को
selection
भर
and
पेट,
isolate
जिसने
individual
ना दी
cell
माँ बाप को भर
components.
पेट रोटी कभी
Please
जीते
note
जी,
that the
आज
drug selection doses need to be determined by kill
लगाते
curve
उसको
for each
भंडारे
cell
में
line.
मरने के बाद देखा।
5 days after puromycin selection of transfected HEK293
अब
cells,
ये सही है,
almost all cells in the 2 control transfections died. However,
लदी
as
हुई है
expected,
रेशमी
the
चादर
co-transfection with the
वो हरी
gRNA
मजार,
construct produced puromycin-resistant
लदी
cells.
हुई है
The
रेशमी
fourth
चादर
step
वो हरी
is to
मजार
analyze the
से वो
puromycin
हरी
positive
मजार पर
cells to detect the
बाहर एक
knockout.
बुढ़िया माँ को
If you
ठंड से
have a good
छुटते देखा।
protein-specific antibody, you can use
चादर
Western
चढ़ा के
blotting
गए
to
वो
evaluate the knockout. It is best to use this method after
आत्महत्या
isolating
कर ली
single
क्रिकेट ने
colonies.
सुसाइड नोट छोड़
Another
कर, अब
method
इंसान
is genomic
से
PCR
ज्यादा
to
रंग
verify the
बदल नहीं
correct
सकता।
GFP puromycin integration in the genome, followed by
अभी इसका मैं क्या
sequencing
करूँ,
to
वीडियो बना
confirm
देखिए,
the
मैंने एक बहुत
integration.
बड़ा काम कर दिया
To
क्या?
amplify only the edited allele, design PCR primers following these rules. The forward primer should be upstream of the 5' end of the left homologous arm of the donor vector, and the reverse primer should be in the GFP region. In this example, knockout ATG5 gene was conducted by the protocol. Genomic PCR products of the correct size were amplified from the gRNA constructs, plus the donor co-transfected cells, and not from the scramble control transfected cells. The PCR products were further sequenced using the PCR primers to confirm the correct integration. Sequencing data using the forward primer shows the correct integration of the 5' end of the left homologous arm in the donor vector. The reverse sequencing primer confirmed the correct integration of GFP puromycin cassette. The original ATG5 gene was replaced with GFP. Origene offers genome-wide human and mouse CRISPR knockout knock-in kits. With these complete, ready-to-use kits can knock out almost any gene from the genome and knock in the GFP and puromycin cassette simultaneously. You can study the consequences of the gene disruption and potentially do a promoter study at the same time. Origene also has other functional cassettes to provide different fluorescent proteins, luciferase, or different selection markers. If you wish to have the CRISPR knockout kit created a different donor vector, please visit our website to request a custom project. To learn more, please visit the